Hi everyone, welcome to the AIX Biology channel. Today we are going to look at a 2026 review paper that should make anyone who reads the food labels feel a bit uncomfortable. There is a small molecule called polyphosphate. It is used everywhere as a stabilizer and emulsifier in processed food. There is emerging evidence suggesting that it may also drive the molecular machinery of Alzheimer's disease and other types of tauopathies. So let's dive into it and see what the case actually looks like. This is the paper's graphical abstract. On the left, you have a plate of everyday food, and on the right, you have a neuron filled with neurofibrillary tangles and disintegrating microtubules. These are the molecular hallmarks of Alzheimer's disease. The molecule connecting them in between is the polyphosphate, joining the center as a chain of phosphate groups. The author's core argument in this paper is that this molecule, used as a food additive that almost no one thinks about, may act also as a biochemical accelerant for tau aggregation inside dying neurons. The rest of the paper is trying to build the case. Tauopathies are a family of neurodegenerative diseases that look very different on the surface. Alzheimer's disease is mainly a memory disease. Frontotemporal dementia shows up as personality and language changes. ALS is a motor neuron disease, but peer into the brain tissue under the microscope and you will see the same thing in all of them. And that is the clumps of a protein called tau that have aggregated into insoluble neurofibrillary tangles. The tau protein normally stabilizes the microtubule highways inside neurons. When tau misfolds and aggregates, those highways collapse and the synapses fail and the neurons die. So even though these diseases may have different clinical phases, they really share the same molecular culprit. To understand why polyphosphates are important in this context, we need to have a quick look at tau itself. Panel A here is showing the architecture of tau. It's a long disordered protein with proline-rich domain and then 4 repeats called R1 through R4 that form the microtubule binding domain. And the microtubule binding region is rich in lysine, which are positively charged amino acids highlighted in green. There are 2 short hexapeptide motifs. We call them PHF6 and PHF6 star. They are the nucleation triggers, and these are the segments that flip into a beta strand conformation and start the aggregation cascade. Panel B is the atomic structure of the filament core, showing all these side chains packing together. Panel C illustrates how 2 tau monomers link to each other at a small 3-glycine patch, the GGG patch, to start oligomerization. And panel D is the electron microscopy image for these tangles. So the takeaway is tau aggregation hinges on a small number of hotspot amino acid residues, and anything that exposes them can be dangerous. Tauopathies are not a linear failure. They are a self-reinforcing cycle. It starts when tau gets hyperphosphorylated, which makes them detach from the microtubules that they are supposed to stabilize. Once free, tau aggregates into oligomers, then into fibrils, and then into tangles. Those aggregates block axonal transport, and that includes the transport of mitochondria, which means the synapses are going to be starved of ATP. The damaged mitochondria leak reactive oxygen species, and the reactive oxygen species can feed right back into the kinases that hyperphosphorylate tau in the first place. So you see, this balance is not stable. Once it tips, it accelerates, and any molecule that can change or shift this balance even slightly is a potential disease driver. And this is where polyphosphate polyphosphates come in. Polyphosphates are simply chains of inorganic phosphate units linked by phosphoanhydrate bonds. They are conserved from bacteria to humans, and they come in three rough size classes. The short chain ones, which are 3 to 10 units long, include ATP, which is the cell's energy currency. The medium chain ones live in the mitochondrial matrix, and they are the chelating agents with ions. The long chain ones, which can be up to 1,000 units long, are found in inflammatory contexts, and these are the ones most strongly implicated in protein aggregation. So this brings the twist this review hammers on. Polyphosphates are not only made inside our cells, they are also one of those most commonly used food additives in the world, used to emulsify cheese, to stabilize processed meat, to adjust pH, and so on and so forth. The point is, most of us eat them every day. This is the paper's main figure to show what goes wrong in a dying neuron. On the left, you see the tau cascade, undesired post-translational modifications, then tau forms oligomers, and then bigger filaments, and eventually the big aggregates that can propagate from neuron to neuron in a prion-like way. Right here, it shows a healthy neuron with its axons and synaptic terminals. On the right side, it shows what happens in the synapse during disease. Tau and amyloid beta aggregates, and they impair the transport of mitochondria along the axon. The mitochondria that do make it are damaged. ATP synthase is impaired, reactive oxygen species accumulate, and the synapse runs out of energy. This is the cellular picture of synaptic failure that translates into memory loss and cognitive decline in the patients. Figure 3 shows how the authors think polyphosphates can contribute to the story. On the top, you see a tau monomer with its domain color-coded. The positive charged regions are marked with plus signs, and then the polyphosphates are the blue spheres carrying negative charges. In step 1, the negative polyphosphate binds electrostatically to the positive tau, particularly in the microtubule binding repeats. This is a direct interactions between them. And in step 2, the binding can induce a conformational arrangement. Tau, which is normally floppy and intrinsically disordered, gets pulled into a conformation-prone shape that exposes the sticky motifs and the 3 glycine patch. In step 3, which is on the right, that exposed conformation now finds another tau molecule, and the triple glycine patches will lock together with each other. And now you have the start of an oligomer. Beyond direct binding, the polyphosphates also lower the kinetic energy barrier to nucleation, and they modulate the kinases that can phosphorylate tau, so the 3 mechanisms all point towards more tau pathology. It's important to keep in mind that the polyphosphates don't only act through tau, they also hit mitochondria directly. In healthy cells, polyphosphates buffer calcium, modulate reactive oxygen species production, and support respiration. But in disease conditions, these regulations break down. Polyphosphates accumulate around mitochondria, drive calcium overload in the matrix, and trigger opening of the mitochondrial permeability transition pores. Once that the pore opens, the membrane potential is going to collapse, and cytochrome C can leak out. ATP synthase is going to be stopped, and electron transport chain starts leaking electrons that become reactive oxygen species. The cell is now on the path to apoptosis. And because reactive stress also drives tau hyperphosphorylation, you are back to the vicious cycle that we have seen a few slides ago, except that now the polyphosphates are accelerating both ends of it. And here comes the most uncomfortable part. This figure summarizes the everyday food uses of polyphosphates. They are in bread, in processed meat, in cheese, in pizza, in juice, and so on and so forth. And in Western diets, total phosphate intake from these sources can be 1 to 2 grams a day. And this amount can be much higher in the consumers who consume a lot of heavy processed food. The triangle here in this figure is particularly interesting because it raises the concern that accumulation of polyphosphates may cause different consequences, such as oxidative damage stress, inflammation, or even blood-brain barrier concerns. The food industry has used those compounds for decades on the assumption that they are safely degraded and excreted. The authors of this review paper argue that this assumption deserves a hard look, especially for the people most vulnerable to neurodegeneration. Here's the nuance. In healthy young adults, 80 to 90% of dietary polyphosphate is degraded in the gut and absorbed as harmless free phosphate, and the kidneys can flush them out. The intact blood-brain barrier also does not allow polyphosphates to come into the brain. So for most of us, most of the time, this is not a problem. But 3 things can change with age and with disease. First of all, people who consume large amounts of processed food can push their daily phosphate intake to a concentration that is way above what is normal. Also, the blood-brain barrier becomes leakier over time, leakier with age and with certain diseases, so the polyphosphate themselves can be a problem that will disrupt the endothelial barriers. And finally, the function of our kidneys can also decline over time, so excretion is going to be impaired. Unfortunately, the aging population or the elderly population actually have all 3 together at the same time. And this is exactly the population the authors say we urgently need to study. There are a few things we want to take home from this paper. The first one is to realize that endogenous polyphosphates, the ones our own cells make, are emerging as drivers of both tau aggregation and mitochondrial dysfunction, the 2 big hallmarks of tauopathies. The second thing is that these effects form a self-reinforcing cycle that does not easily reverse once it starts. The third thing is that the systems that normally protect us from circulating polyphosphates, for example, gut breakdown, excretion by the kidney, or the blood-brain barrier, they're all weakened over time with aging and with disease. So these findings raise a very important question. Can the polyphosphates we eat every day in cheese, in bread, and in processed meat actually amplify neurodegeneration in the people whose protective systems are already compromised? The author's answer to that question is yes, but I have to say this is a review paper, not a clinical study, so they're not really presenting their data. But they do argue that this question is too important to leave unanswered. There are 2 big implications here. The first one is about food safety and about what kind of regulations we need to protect those people who are specifically and especially vulnerable to neurodegeneration. The second one is about research. The research questions are very clear. We need to actually quantify the polyphosphate concentration in patient brain tissue and do real biodistribution studies. Equally important is to use iPSC-derived neurons to study if polyphosphates can actually accelerate the aggregation of tau proteins in neurons. So This concludes everything I want to share with you today from this paper. If you like this kind of analysis, please click like, and here's more to come. Thank you.